Hello, everyone. This is going to be a review for genetics for the first exam. This is going to just be like a test run to see if you all find this useful or, if there's gaps and you're learning from this. So hopefully this will be a good general review. But if you have any questions or ideas to make it better, please let me know. And I'm going to try to go over kind of everything that we learned, with special attention to the questions that we've reviewed, the kinds of topics there, because I think that would be good to focus on for the exam. So starting off with the things we talked about um, at the beginning of the semester, general things to remember. So 22,000 to 25,000 genomic loci in humans. And the human genome is 3.2 billion bases long, but most of this is non-coding DNA. Essentially, it's just filler. So you have a gene proper region of the DNA, which is where you get the protein from. It codes for protein. But then you also have the cis regulatory switches, which control the when, the where, and the how much of the protein production. So when does it occur in your lifetime? What kinds of cell types is it produced in? And then how much of that protein is produced? And since we focus heavily on humans, we are diploid, meaning we have 2 alleles per locus. And it's important to keep in mind the central dogma, DNA to RNA to protein. So I've made a review here for you. So I have a DNA sequence here. I have the sense or coding strand, and then I have the antisense and template strand. So remember that the template strand is what the mRNA matches to during transcription. But a quick way to also get your mRNA sequence is to just take the sense strand, which is the 5' to 3', and just replace all of the T's with U's with uracil. But it also might be helpful if you just write everything out as well. So in transcription, we're taking that DNA sequence and we're turning it into messenger RNA. And then in translation, we'll have our tRNA match with our mRNA sequence. And then from that, we'll produce amino acids. And in that chain of amino acids, we will have our protein. And remember that for proteins, we always start off with the amino acid methionine, and we always end with a stop codon. More things to keep in mind. So any 2 genomes from the human population have base differences every 1,000 bases, or we would call those single nucleotide polymorphisms, SNPs. So 3 million total, which is a lot, but there is degeneracy in the genetic code. Remember that big table with the nucleotide sequences corresponding to amino acids. You could have several different codons that would translate to the same amino acid. And that's important as we go on and talk about mutations. And so here's our anatomy of a typical genomic locus. So like I mentioned, on your DNA strand, you're going to have some cis regulatory switches, which are non-coding. So they don't code for protein. They don't go through the central dogma above, but they do give instructions to the protein coding region again. So how much protein, when is the protein produced, and where in the body is it produced? And then you have your trans sequences, which is the coding region that goes through the central dogma. It produces protein, And we call this the trans factor because the proteins are transitory. The whole purpose of making proteins is that throughout a cell, it has a lot of different needs. So it needs a good variety of proteins to do the job. So these proteins can move around the cell, go to where they need to go to fulfill a specific function within the cell. And more terminology, homologous loci. So this is just a little picture of what it means to be homologous. So you have one chromosome from your mom, one chromosome from your dad, and these do not have identical genetic sequences, but they are the same kind of chromosome. So here I have chromosomes for number 2. Remember, they are a pair, so they're homologous. They go together. They have the same loci in the same locations, but you get different alleles. So you get, in this case, a dominant allele from your mom and a recessive allele from your dad, making you heterozygous for that specific trait at that locus. And a big thing is loss of function versus gain of function. So loss of function is when there's not enough protein made, or it's not made during the proper times in your life, or it's not made in enough cell types. And when we talk about loss of function, we're talking about autosomal recessive diseases. The two that you need to know for the exam are cystic fibrosis and sickle cell anemia. And in this case, the capital letter is the normal allele. The lowercase letter is the loss of function allele. And remember, to be heterozygous, you are phenotypically normal. You can get by with one loss of function allele, but you are a carrier of that loss of function allele. So in this case, being homozygous dominant is phenotypically normal, you're not a carrier, and homozygous recessive is affected. So you're affected by the autosomal recessive disease because you have 2 loss of function alleles. So you need both of the loss of function alleles to have the disease. On a pedigree, you could have 2 phenotypically normal parents that have a child with the disease, and then you would know that they would be carriers. So something to note when you're looking at pedigrees is if you want to figure out whether it's an autosomal recessive or autosomal dominant disease that you're looking for in the terms of the pedigree, you could look for whether an affected child has 2 unaffected parents, because remember, in a pedigree, it tells you the phenotype, not the genotype. Here on this example pedigree, I've written in the genotypes, but you would have to figure these out on your own. So of course you need to figure out the pattern of inheritance. Is it autosomal recessive or autosomal dominant first, and then you can figure out the genotypes, which will help you figure out probabilities. So in this case, I've designated a capital A as a normal allele, and a lowercase a is a loss of function allele. And so again, the parents 1-1 and 1-2, they have to be both carriers, because you'll notice individual, and I'll write this in for you. So this person is 1-1, and then this person is 1-2. So phenotypically, they're not affected, by this disease, again, either sickle cell or cystic fibrosis. But you'll notice individual 2-2 is affected. So we know that to have an affected child um, and to have phenotypically normal parents, that means that the phenotypically normal parents have to be carriers. And I've given you different examples of breeding combinations that can produce different kinds of offspring, whether they're phenotypically normal but not a carrier, phenotypically normal and they are a carrier, or if they are affected. And I've done the Punnett squares for you as well to show you the different probabilities here. And I highly recommend every time you're looking at a pedigree that you just try to draw some simple Punnett squares just to make sure you understand how the alleles can be passed on and what it means for the phenotypes of the offspring. And here I've given you an example problem. So it says 2 carriers of cystic fibrosis have children. What is the probability of having 2 phenotypically normal children in a row who are not carriers? So this is a probability question. And so the first thing that I would do is I would draw a Punnett square like I've done. So we know both parents are carriers, meaning they are heterozygous. Especially because we know that cystic fibrosis is autosomal recessive. Remember, it's important to note the pattern of inheritance, because then you know that the carriers are phenotypically normal, but they can pass on the loss of function allele to offspring. So here again, capital A is normal, lowercase a is loss of function, and I drew a Punnett square. And because the question specified it wants phenotypically normal children, I got rid of the affected square. So then, so we have in the Punnett square, we have, if we didn't get rid of the affected square, we would have 3 out of 4 gametes being phenotypically normal. So we're looking at the probability of being a carrier within that 3 out of 4. So I got rid of the affected square. And so we are wanting the phenotypically normal children who are not carriers, meaning that they have to be homozygous dominant. And so it seems like they're in the squares we have remaining, we have 1 out of 3 that are not a carrier. So you have a 1/3 chance of being phenotypically normal and not being a carrier. And since we want the probability of having 2 phenotypically normal children in a row who are not carriers, the probability of each individual event is the same for both. So we multiply those probabilities and we would get 1/9. And then just a few other probabilities. So if we would want the probability of having 2 phenotypically normal children in a row who are carriers, we can see, since again, we specified we want phenotypically normal, this time it's a 2/3 chance. It's a 2/3 chance for each. So we multiply those probabilities and we get 4/9. And then if we want the probability of having 2 children in a row who have cystic fibrosis, this time we don't get rid of the square. So there's a 1 in 4 chance of having cystic fibrosis for each child. So we multiply those probabilities and we get 1/16. Okay, so just remember, for autosomal recessive, you need to know that cystic fibrosis and sickle cell anemia are autosomal recessive. diseases, and that if you see two phenotypically unaffected individuals who have an affected child, you know that you're working with an autosomal recessive pattern of inheritance. Um, so here is another pedigree problem about cystic fibrosis, and I believe that this one um, was one of the ones on Blackboard. I thought this one was pretty tricky, so I thought I would go through it. So the question asks, if individuals 5 and 14 breed, what is the probability that they will have a child with cystic fibrosis? So I have labeled individuals 5 and 14. And you can see that they are both phenotypically normal, which means we're going to have to figure out their genotypes. So starting off with individual 3-5, so they're phenotypically normal, but you'll notice that their sister, number 6, is affected. And you'll also notice that both of his parents are phenotypically normal. So that means since we're working with an autosomal recessive pattern of inheritance, they both must be carriers. And this is the same with individual 14. You'll notice they have a sibling who is affected, and both of their parents are phenotypically normal, meaning that they must be carriers. So individuals 3-5 and 3-14 are phenotypically normal. So to have a child with cystic fibrosis, they would both need to be carriers. So I've drawn that a Punnett square. So because we know individual 3-5 is phenotypically normal, we're looking at their chance of being a carrier. So I've gotten rid of the affected square. So the chance of 3-5 being a carrier is 2/3, and it's the exact same for individual 3-14. So what we're doing here is we have to assume that all of these outcomes are true. We have to assume that 3-5 is a carrier, and the chance of that being the case is 2/3. We also have to assume that 3-14 is a carrier, which is also 2/3. And then we would cross these individuals, and it's the same Punnett square here, just without the affected square being crossed out. So the chance of having two carriers producing a child with cystic fibrosis is 1/4. Therefore, in this case, the probability of 3-5 and 3-14 having a child with cystic fibrosis is 2/3 times 2/3 times 1/4 equals 1/9. And again, we have to assume that all of these outcomes are true. So we have to assume, because we know that 5 and 14 are phenotypically normal, we have to assume that they are carriers to even consider the probability of them having cystic fibrosis in the first place. And so we use those individual probabilities to find the probability that we're looking for. So this one I thought was kind of tricky because there's a lot of pieces that you have to consider. So if you have questions on this, don't hesitate to ask. And again, this is on Blackboard. So moving on to autosomal dominant, which is definitely a different pattern of inheritance. The two kinds of diseases that you need to know that are autosomal dominant is Huntington's and achondroplasia. So for autosomal dominant, we're working generally with gain of function. So remember that the genotype of a normal individual in an autosomal dominant pattern of inheritance is homozygous recessive, so 2 little letters. An affected individual is heterozygous. And this is because being homozygous dominant for an autosomal dominant pattern of inheritance is embryonically lethal, meaning that we don't have any live children. So they are not able to breed. And a key factor for autosomal dominant pedigrees that you need to note is that every affected child has an affected parent. And this goes back to the fact that affected individuals are heterozygous. So again, I have labeled um, the genotypes on this pedigree for you. Um, it's pretty quick to do them yourself. I would recommend you just go ahead and label them if it's not, a huge pedigree. And then I've also done just some examples of different crosses you could have, and then the Punnett squares. And here's an example problem for you. It says Bob's parents both have Huntington's. What is the chance if he marries someone who does not have Huntington's that he will have a child with Huntington's? So remember, Huntington's is autosomal dominant. And so we know both of his parents have Huntington's. So that means that they have to both be heterozygous, because if they were homozygous dominant, they wouldn't be alive to have children. So I've drawn a Punnett square here for both of Bob's parents. Again, both heterozygous, and I crossed out the homozygous dominant square because there will be no live birth. And we know Bob is clearly alive and walking around. So there's a 2/3 chance of Bob having Huntington's. And so we kind of have to assume that this is the case, just because we're considering the probability of him having a child with Huntington's. And we know he's marrying somebody who does not have Huntington's. So he kind of has to have Huntington's to have a child with Huntington's, if we know that his partner does not have Huntington's. Yeah, so for Bob to marry someone without Huntington's and also have a child with Huntington's, he must have Huntington's. So therefore, Bob's genotype would be heterozygous. And so we're crossing him with somebody who does not have Huntington's. They have to be homozygous recessive. So the chance that his child would have Huntington's is 50% here. So we have to assume that he has Huntington's, and there's a 2/3 chance of that. And so there's also a 50% chance that his child will have Huntington's. So 2/3 times 1/2 equals 1/3. So what you have to remember for these more complex probability problems is that you have to assume that certain things are the case. Um, like you have to assume a particular genotype. And it's really just based on what the problem gives you. So we knew that both of his parents had Huntington's, and we know that they have to be heterozygous. And since he's marrying somebody who doesn't have Huntington's, they would be homozygous recessive. So yeah, it's just kind of like a process of elimination. Um, for him to have the potential to have a child with Huntington's, he would have to be also affected. And then just some mutations we talked about for loss of function and gain of function. So for loss of function, in the trans region of the DNA, you could have a missense mutation, a nonsense mutation, and a frame shift. So remember, missense mutations are a point mutation where an amino acid sequence is affected. Well, one amino acid is affected at least. A nonsense mutation changes a normal codon to a stop codon. So you get a non-functional protein because you're cutting off the amino acid sequence before it could chain to get the protein that you're trying to make. And then a frame shift is an addition or deletion of nucleotides of a number that's not divisible by 3. And so because codons are read as 3 nucleotides, everything downstream of that mutation site, it would have its sequence affected. So you can think of it as like in like in a sentence, like if I wrote, my name is Callie, and if I were, we're keeping essentially the length of the words the same. And again, because codons are 3 nucleotides, if I were to like insert like a C, then it would be MC, YNAMEI. Like it's just everything downstream of the mutation site is just, it's just garbage essentially. And so other mutations for gain of function, again, missense, an addition, or a duplication. And there can also be mutations at the cis regulatory switches, which would affect the amount of protein being produced, whether there's a mutation causing too much or too little, if it's not happening at the correct time in development, or if it's not happening in the proper cell types. And so a few more definitions. We're getting to the end. Mendel's law of independent assortment. This is the law that says that alleles sort independently during gamete production. So when they are being separated, their order just kind of random when they come together. Like there's no rhyme or reason to it. They just kind of all are slapped together, and then what the cell and the gamete ends up with, it just, that's what it ends up with. So that's how you have genetic variation. And another similar law by Mendel is his law of segregation, saying that alleles separate or segregate during gamete formation. So again, there's like a randomness to how they're sorted. And then we know that they're also separated. So there's quite a few ways for the gametes to have genetic variation, crossing over, and then the way that they're arranged when they get to the metaphase plate, and then remembering that they are going to be separated and pull apart during anaphase. And lastly, a few more things that you need to know. We talked about non-disjunction and chromosome number. So aneuploidy is when you have like an extra or a missing chromosome. So essentially you don't have the normal amount of chromosomes. Monosomy is when you have one chromosome missing, and trisomy is when you have an extra chromosome. And we talked about the shorthand that you need to know for the exam. So 46, XY is a normal biological male. 46, XX is a normal biological female. Then you need to know Turner syndrome, Klinefelter syndrome, Jacob syndrome, and Down syndrome. Turner syndrome is when the individual only has one X chromosome. So it's 45, X, 45 because they're missing a chromosome. And then we know that they just have one X chromosome. Klinefelter syndrome, they have an extra X chromosome. So it's 47, XXY. Jacob syndrome is when they have an extra Y chromosome. And then trisomy 21, remember trisomy is an extra chromosome. So you have 3 copies of that chromosome instead of 2. So it is 47 because we have an extra chromosome, XX. This just says that we have a female, a genetic female, and then plus 21, that designates that we have an extra chromosome 21. So we would have 3 chromosomes at chromosome 21. So how does aneuploidy happen? The mechanism in which it happens is non-disjunction, which results in daughter cells with aneuploidy. So non-disjunction is when the chromosomes or the chromatids fail to separate. And there's a couple of forms of this. We focused primarily on meiosis, but it can happen in mitosis. But we're talking about these diseases that can be that can be caused by non-disjunction. So we're focusing on meiosis. So in meiosis one, if we would have non-disjunction there, this is during anaphase one, so the homologous chromosomes fail to separate. And this is worse than non-disjunction in meiosis two, because if you think about it, the earlier on that problems happen in meiosis, the more subsequent steps that you're going to have like problems occurring in, kind of like, because if you create a problem early, like your whole thing afterwards is going to be affected. Like you're going to have the incorrect amount of chromosomes um in the cells, like when you go through meiosis 2. So it's worse than non-disjunction in meiosis 2. And meiosis 2, the chromatids fail to separate. Remember at this point, there's crossing over. So, I mean, we don't really call the chromatid sisters, but they are chromatids. So they fail to separate during anaphase 2. So you have a cell that has too many chromosomes now, and then you have a cell that doesn't have enough chromosomes, which is bad, because then you're spreading that affected gamete to your offspring. And again, like I mentioned, non-disjunction can happen in mitosis, but we're not really focusing on that. So that was just a general review. If you have questions, let me know. Don't forget to go over all of the practice on Blackboard. There is a very detailed study guide there, but I just kind of wanted to provide a general overview. So if you have any questions, concerns, if I got something wrong, because we're all in this together, you let me know.